Assalamualaikum and hi everyone. Today we will continue on chapter 5, biological molecules, on another two biological molecules that we haven't touched yet, which are proteins and also nucleic acids. So in this chapter, later, you will learn about how does the protein form, and also how does the nucleic acid form, and what are the important components inside the proteins and also the nucleic acids. So what is protein? Protein is a biological molecule that consists of one or more polypeptides, meaning to say that a protein is not always consisting of one polypeptides only. So protein molecules are made up of hydrogen, carbon, oxygen, nitrogen, and also other elements. So polypeptides, as you remember, are the polymers built from the same set of 20 amino acids. It's meaning to say that amino acid is the monomer for polypeptide, and the polypeptide is the polymer structure of the protein. And amino acid, which is the monomer, are molecules with carboxyl and amino groups. And also, all amino acids have different properties based on its side change, or also known as the R group. So let's see the structure of the amino acid monomer of the protein. So here is the example of a polypeptide change. We have one amino acid over here, second amino acid, third and fourth amino acid, in which all of this is linked together by a peptide bond through dehydration process. And this is the amino acid structure, the monomer of polypeptide. As you can see here, we have a double carbon backbone, and here is the amino group NH2, and the other side we have carboxyl group here, COOH, and this one is referred to the side change, or known as the R group. So all of the amino acid consists amino group and also carboxyl group. Bear in mind, the only difference between all amino acid is referred to the R group, or the side chain. So there are 20 amino acids. Here we have amino group, and then this one is the carboxyl group. Here is the carbon backbone, and this is the side chain. As you can see in the orange box here, showing the side chain of each amino acid. So here we have glycine, or always notated by G, alanine, valine, leucine, isoleucine, proline, tryptophan, phenylalanine, methionine, and we have another serine, threonine, cysteine, glutamine, asparagine, and tyrosine. So that is all of 24 molecules of amino acids. And another one, we have aspartic acid or aspartate, glutamate, lysine, arginine, and also histidine. So how does the polypeptide form, or the protein form? So a polypeptide is a polymer of amino acid, and it was linked by a peptide bond through dehydration process. Remember, all monomer becoming a polymer through a dehydration process, loss of water. And the polypeptide length varies from a few to more than 1,000 monomer, and each of the polypeptide is a unique sequence of amino acid with carboxyl N, the C terminus, and it also has an amino N, which is the N terminus. Okay, here is the N terminus, and here is the C terminus. So these are two amino acids. Okay, first monomer, second monomer, meaning two amino acids linked together by a peptide bond, and these molecules can bind or can be bonded to another monomer or another amino acid through dehydration process. So new peptide bond will form between these two monomer, and it will become another molecules of peptide. So this is another example of how does the peptide bond formation through dehydration process. One monomer, loss of the water, and then the peptide bond will form over here. So the protein structure, it depends on its shape. So the protein will have its function, and you have to remember that one protein consists of one or more than one polypeptides. So the structure sometimes it twisted, folded, and coiled into a unique shape. So a protein structure is the one that responsible to determine what is the functions of the protein. And this structure is due to the sequence of amino acid. So let's see over here. You can see a different structure of protein, and there are 4 levels of protein structure. Have primary structure, secondary structure of protein, tertiary structure, and last but not least is quaternary structure. So primary structure is actually a sequence of change of amino acid. Remember, amino acid is the monomer. This one ball over here is referred to one amino acid, second amino acid, third, fourth, 5, 6, and until at the end of it. And all of these amino acids are linked together by a peptide bond. So it's like a chain of amino acid. And next we have secondary structure of amino acid. This secondary structure form when the peptide backbone interact with each other through hydrogen bonding. So hydrogen bonding is the only bond that responsible to the secondary protein structure. Okay, so it can form beta plated sheet structure or alpha helix structure. Next, we have tertiary structure. Tertiary structure is the 3D structure of a protein in which it involves lots of types of bond here, and this interaction or this structure is due to the interaction between the side chain. Okay, secondary structure is due to the peptide backbone, interaction of peptide backbone. Meanwhile, the tertiary structure is due to the side chain of each of the amino acids. And last one, we have quaternary protein structure. Quaternary protein structure consisting of one or more amino acid chain, in which, as you can see here, this is the first tertiary structure. It combined with another tertiary structure here, and thus forming a quaternary structure. But bear in mind, not all protein consists only one or two polypeptide. It can consist more than that. So this is the clear view of the primary structure, sequence of amino acid of the protein bonded by a peptide bond. And then secondary structure is the coil form of protein, which is known as alpha helix, and also it can form a folded structure, which is known as beta plated sheet. And this structure is caused by the interaction between the peptide backbones through hydrogen bonding. Okay, as you can see over here, alpha helix and beta plated sheet structure. Tertiary structure is caused by the interaction between the side chain or the R group of the amino acid. And these interactions include hydrogen bonds, ionic bond, hydrophobic interactions, and also van der Waals interaction. And sometimes disulfide bridge also may reinforce the protein structure. Okay, here hydrogen bond, hydrophobic and van der Waals. We have disulfide bridge and also ionic bond. This is interaction between one side group to another side group. Okay, here tertiary structure. And then last one, we have quaternary structure, in which it results when two or more polypeptide chain to form a molecule, macromolecule on one protein, as you can see here. But bear in mind, sometimes a protein also can form only from one polypeptide chain. Okay. So examples of the protein that have a quaternary structure are collagen that you can find on your skin, and also hemoglobin is also example of the protein that have a quaternary structure, where you can find it in your red blood cell. Okay, this is the picture of hemoglobin. And protein can change its shape through the process of denaturation and also renaturation. So what does it mean by denaturation and renaturation? Denaturation is the process that changes the shape of the protein without breaking its peptide bond, meaning to say denaturation only break the hydrogen bonds, the sulfide bridge bonds, and other bonds other than peptide bonds. Meanwhile, renaturation is the vice versa of denaturation process, in which we want to fold back the protein into its original shape. Okay, it's meaning to say from a single change of amino acid, we want to make it form or folded back into secondary, quaternary, or tertiary structure. So, when denaturation occurs, what will happen? The protein will lose its biological activity, because the protein that have quaternary structure or tertiary structure are the one that only have function. And then if you have denatured protein, you can regain back its activity, okay, through renaturation process, and it will becoming a new tertiary structure, quaternary structure. Okay, meaning to say it becomes a normal protein back. So this is the example of denaturation process that cannot be turned back, such as frying an egg. When you fry an egg, the fried egg cannot back to become a whole single egg. However, a protein such as milk, okay, when you heat it, it will become inactive protein. However, when you call it back, it can back to its original shape. Okay, therefore, this process is known as renaturation process. So there is a lots of method or agent that can be used to denature the protein, such as the heat, mechanical treatment such as beating the egg, and also using a very high or a very low pH, such as you adding a lemon juice inside a milk, so the milk will become sour, and sometimes it forming a curd whey or yogurt. So, what is the functions of the importance of the protein? There is a lot of functions and importance of protein. We're going to look at it one by one. The first one, protein is very important for support, in which you can find protein in such as the keratin protein inside your hair. Okay, and the collagen also, and elastin, you can find it on your skin. And protein also responsible for the storage of the amino acid. For example, casein is a protein that inside the milk. They're responsible for the source of amino acid for baby mammals. And inside the egg white, we have ovalbumin protein, okay, which is this is important source for the developing embryo. Transport protein, this one you can found in plasma membrane, such as the transport protein, integral proteins, and other transport protein. And then protein also responsible for respond to the cell in chemical stimuli. This one happen in your nervous system, and this occur in the synapse where the neurotransmitter will bind to your receptor protein and then transmit the nerve impulse from one synapse to another, from one cell to another nerve cell. And then it also responsible for the coordinations of your organism activities. This is known as a hormonal protein such as the insulin. This one is responsible for regulating the blood sugar concentration. And then we have contractile and motor protein. This one is very important for movement, such as the actin protein and also the myosin protein. And then we do also have a defensive protein that is responsible to protect you against the disease, such as the antibody. Enzymatic protein. This one is the protein that make up the enzyme. Functions of the enzyme is a biological catalyst that speed up the reaction without changing at the end of the reaction. This one you have learned it when you are in semester 1, right? Okay, next let's look on nucleic acid. So, nucleic acid is another example of biological molecules in which it is a large molecule consisting of long chain of monomer that called as the nucleotide. So, meaning to say, the nucleotide is the monomer for nucleic acid and its polymer form is known as nucleic acid or polynucleotide. So examples of nucleic acid are DNA and also RNA, ATP, adenosine triphosphate, also another example of nucleic acid. So what is the component of the nucleic acid? So nucleic acid are the polymer which is known as a polynucleotide, and each of the polynucleotide is made of the monomer called as nucleotide here. And each of the nucleotide, the nucleotide monomer, it must consist of a nitrogenous base, a pentose sugar, and one or more phosphate group. As you can see here, we have nitrogenous base over here, pentose sugar, and also we have a phosphate group over here. So the portion of a nucleotide without the phosphate group is called as nucleoside. Okay, if you add phosphate group over here, it is known as a nucleotide. So in DNA, the pentose sugar is deoxyribose, while in RNA structure, the pentose sugar is a ribose sugar. So let's look here. So here, this one is a nitrogenous base. We have different nitrogenous base: adenine, thymine, guanine, cytosine, and uracil. And it is connected to the first carbon of pentose sugar. Remember, pentose sugar is a sugar that have 5 carbon atom at the backbone. So you have first carbon over here, second carbon, third carbon, fourth, and the fifth carbon. So pentose and bases will make up a nucleoside. And then when you add a phosphate group here, and all of this structure will known as a nucleotide. which is a monomer for polynucleotide or a monomer for nucleic acid. So let's look at the polynucleotide or nucleotide polymer. Nucleotide polymer are linked together to build a polynucleotide through dehydration process, which is loss of water, in which the covalent bond is formed on the OH group on the third carbon of first nucleotide to the phosphate group on the fifth carbon on the next. So let's see, what does it mean? Okay, let's look over here. This one is a phosphate group that link to the fifth carbons of the sugar. Meanwhile, this nucleotide will link to another nucleotide over here. You just imagine, and the third carbon will link to the phosphate group of another nucleotide. So it will become a long chain of nucleotide, and it will becoming a nucleotide or nucleic acid. Meanwhile, here the orange one is referred to the nitrogenous base. This is only one strand of polynucleotide. And the nitrogenous base, which is referred to the orange one, they are differentiated into two families. First, we have pyrimidines. Pyrimidine consisting of cytosine, thymine, and uracil, in which thymine you can only found in DNA, uracil you can only found in RNA, and their structure have single six membrane. I will show it to you later. Meanwhile, family of purine consists of adenine and also guanine. So let's look at the structure of pyrimidine and purine. So as you can see over here, purine family, the structure is only one single six membrane. Why it's called a six membrane? Because it has six angle over here. So 1, 2, 3, 4, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Meanwhile, for purine, it consists of 5 membrane fused together with 6 membrane structure. So 1, 2, 3, 4, 5. 5 membrane linked together with 6 membrane. 1, 2, 3, 4, 5, 6. Okay, this one pyrimidine. Okay, and this one the sugar. In nitrogenous base, we have cytosine, thymine, uracil, guanine, and adenine. Meanwhile, sugar, we have two types of sugar, pentose sugar that involved in nucleotide. For DNA, we have deoxyribose sugar. Meanwhile, for RNA, we have ribose sugar. So the difference between these two sugars is that the presence of the oxygen on carbon number 2. Ribose have oxygen on carbon number 2. However, for deoxyribose sugar in DNA does not contain any oxygen at carbon number 2. So, all of this is the nitrogenous bases that you can find in DNA: cytosine, guanine, adenine, and thymine. Meanwhile, this is a list of nitrogenous bases that you can find in RNA, which is cytosine, guanine, adenine, and uracil. Next, after we look on the nitrogenous base, now we look on the pentose sugar. The pentose sugar is a 5-carbon sugar. In RNA, we have a ribose sugar. Here, it has oxygen over here with carbon number 2. Meanwhile, in DNA, it does not have oxygen over here, so it is known as deoxyribose sugar. Okay, let's look on the nucleoside. How does the nucleoside form? Okay, remember, nucleoside only consists of pentose sugar and nitrogenous base. They are linked here, over here, nitrogenous base is linked to the first carbon of pentose sugar through a glycosidic linkage. Okay, so usually the name of the nucleoside have O sign for purine and idine for pyrimidine at the end of the name. Okay. Next, we're gonna look at the nucleotide. A nucleotide is where a nucleoside combine with a phosphate group to become a nucleotide, and the link here is known as a phosphate ester bond, or can be called as phosphodiester linkage. So again, between nitrogenous base and pentose sugar is glycosidic linkage. Between pentose sugar and phosphate group is phosphodiester bond. And this is how the nucleotide form. A nucleotide form when a nucleoside combined with a phosphate group, and it will form a nucleotide by forming a phosphodiester linkage over here. And this is the examples of nucleoside and nucleotide. This one also. And this is how they name it. And so, what is the functions of nucleic acid? Let's look on the functions of the base here first. So the bases, the function is to construct the nucleotide, and then next we look at the monomer, which is the nucleotide. Function is to form the DNA and also the RNA, which is a nucleic acid, and it involved in cell signaling and auto-metabolism. And the nucleic acid polymer function is to store and transmit genetic information, and also to synthesize a new protein. So, I already mentioned this one just now, in which the structure of the nucleic acid is linked by a phosphodiester bond or phosphodiester linkage. Okay, as you can see over here, this is one molecule of nucleotide. This is another molecule of nucleotide. And then, through dehydration process, these two nucleotides will link together through a phosphodiester bond. Okay? Phosphodiester bond is the one that link between one monomer or another monomer of nucleotide. And this is the structure of nucleic acid. Okay, this is one nucleotide linked with another nucleotide. So here we have 4 nucleotides. 1, 2, 3, and 4. So it always read from 5 prime to 3 prime. So it starts from here, goes down here. Okay, so how does we read it? We read this strand by 5-ACGT3. Okay, 5-ACGT3. And examples in RNA structure. Okay, remember RNA does not have thymine. They only have uracil as a replacement for thymine. And of course, from one monomer to another monomer is linked by phosphodiester linkage. So let's try to read this one. 5 prime ACGU 3 prime. Okay, let's look on the structure of the DNA. DNA have a double helix structure, in which again in DNA they containing bases of adenine, cytosine, guanine, and thymine. They do not have uracil, and all of these are linked together by phosphodiester bond. So let's try to read this one. 5 prime ACGT 3 prime. So what does it mean by a double helix? Double helix is a 2 strands, because DNA have 2 strands that wind together. So how does this 2 strand bonding? It helped by a hydrogen bond. Okay, let's look over here. Okay, this is the first strand of DNA, and this is the second strand of DNA, and they are linked together through a hydrogen bond between the nitrogenous bases. So this is the nitrogenous base of the first strand, and this is the nitrogenous base of the second strand, and they are linked together by a hydrogen bond. Okay, so remember, adenine is always linked by two hydrogen bond with thymine. So this is adenine and this is thymine. They are linked together by two hydrogen bonds. And guanine is always linked by three hydrogen bond with cytosine. We have guanine over here and cytosine. They are linked by three hydrogen bonds. Okay, 2, 3, 2, 3, 3, 3, and so on. And it will become a double strand or double helix structure of DNA. So let's try to complement base this one. So A will bond with T, G with C, T with A, and so on. So you call it as a 5 to 3 prime, 5 to 3 prime. Okay, vice versa. So DNA is responsible for DNA replication process, in which it is a genetic information. This is the way for genetic information to maintain each type the cell divide, in which when the double strands of DNA will break and unwind, one strand will go for the process of translation, meanwhile another strand, it will be used as a template for DNA replication. Okay, and the one that I mentioned just now, the new copy of the DNA will use as a template to make the mRNA, and the mRNA later will use to make the protein through a translation process. So RNA, which is an example of nucleic acid, they have three types. We have messenger RNA, transfer RNA, and ribosomal RNA, in which their function is to transmit the information from DNA to make a protein. So mRNA carries genetic information from DNA to ribosome. Meanwhile, tRNA or transfer RNA bring amino acid to the ribosome to make protein, and ribosomal RNA make up 2 of 3 of ribosome where the protein synthesis take place. So all of this later you will learn further when you in semester 3, okay, in the course of metabolism and cell division. So this is type of DNA and the nucleic acid, okay, is important for your information inheritance called as a gene. And a nucleic acid, the monomer is known as a nucleotide. Okay, then this is how does it look like. The DNA replication occur over here, and then a translation process occur, and this will form mRNA. mRNA will go to the ribosome for the synthesis of the protein. So this is the amino acid. Okay. So the structure of DNA and RNA. RNA is a single polypeptide. Meanwhile, DNA they have two strands of polypeptide forming a double helix. And in DNA, the backbone is always read from 5 to 3 directions, and the another strand is anti-parallel with these ones. Okay, so one DNA molecule includes many genes inside. And the structure of the DNA is double strand, so meaning to say each of the nitrogenous base must have its partner, so that is known as a complementary base pair. Okay, let's see over here. This strand complement base pair to the second strand. This is known as the pairing through hydrogen bond. So, what is the difference between DNA and RNA? DNA, the type of sugar in it is deoxyribose. RNA consists of ribose sugar. DNA have double strand. RNA is single strand. Nitrogenous base that you can find in DNA, adenine, cytosine, guanine, and thymine. Nitrogenous base that you can find in RNA, adenine, cytosine, guanine, and uracil. And then DNA can be found in the nucleus, and RNA can be found in the nucleus and also the cytoplasm. Okay, this is another example. You can read it on your slide later. So I think that's all for today. Thank you, class.